Okay, let's dive in. We all know bananas, don't we? I mean, that perfect snack, breakfast staple. Pretty much everyone loves them.
They're everywhere, yeah. Super convenient.
But here's the thing. What if I told you that very convenience, the fact they don't have seeds, is actually, well, it's a huge problem. That this fruit we love is facing a really serious threat.
It's a bit of a paradox, isn't it?
Exactly, the banana paradox. Today, we're looking into why their genetics are so fragile, making them vulnerable and really difficult to improve through breeding.
But there is some good news on the horizon.
Right. There's been a major scientific breakthrough in what they call banana genomics. It's offering a new lifeline, potentially.
A really significant step forward.
So our mission today, for you listening, is to unpack how scientists are using some incredibly advanced genetic mapping to understand and hopefully, save the banana.
We'll get into the weeds a bit, the challenges which were massive.
Yeah, and the really clever solution they came up with, and what it all means, not just for, your smoothie.
Although important.
Definitely important, but also for global food security and maybe even other crops dealing with similar genetic hurdles.
So you're getting a shortcut today to understanding a really complex issue with global implications. Lots of fascinating stuff here.
Okay, so let's start with that core problem. Why are bananas, this food staple for millions worldwide, actually so fragile?
Well, it's kind of counterintuitive. You see thousands of types of bananas globally, right?
Yeah.
But commercial production, it's almost entirely dominated by just a handful of varieties.
Like the Cavendish. That's the main one we see in stores.
Exactly. The Cavendish is the poster child for this. And this tiny gene pool, this lack of diversity, makes the entire crop incredibly susceptible. Think pests, diseases, climate shifts. It's like putting all our eggs in one genetic basket. And that basket's got some holes in it. It's really quite precarious.
And trying to breed better, stronger bananas isn't simple either, is it? It sounds like a real nightmare for scientists.
Oh, it's notoriously difficult. There are layers to it. First off, most commercial bananas, like the Cavendish, are sterile triploids. Meaning they have 3 sets of chromosomes instead of the usual 2. And crucially, they don't produce seeds. They reproduce vegetatively, like clones.
Convenient for us eaters, no pesky seeds.
Right. But terrible for breeding. You don't get that natural shuffling of genes, that recombination that happens with sexual reproduction. It severely limits the ability to introduce new traits.
So you can't just crossbreed 2 good banana plants easily, like you might with corn or apples.
Not easily at all, no. It's possible, but very complex and slow. And add to that, they have really long growth cycles. It takes ages to see results.
Okay, slow growth, seedless. What else?
Well, here's the kicker. Genetically speaking, their genomes are full of what we call large chromosomal rearrangements.
Right, you mentioned that. It sounds messy.
It is. Imagine the instruction manual for building a banana. But whole sections are cut out, flipped around, pasted in the wrong places. It completely scrambles the inheritance pattern.
So trying to find a specific gene for, say, disease resistance becomes like finding a needle in a haystack that's also been through a shredder.
That's a pretty good analogy, yeah. It makes it incredibly hard to link specific genes to desirable traits.
And I remember you saying standard genetic tools like GW with genome-wide association studies, which have been revolutionary elsewhere, they didn't work so well with bananas because of this genetic jumble.
Precisely. Those powerful methods, which scan genomes to find trait-linked genes, were much less effective. Those rearrangements created too much noise, too many false leads, or just missed signals altogether. It was a huge barrier.
Okay, so the pressure was on. Global food systems facing challenges, bananas being vulnerable. Something had to give.
Exactly. Understanding banana genetics became really urgent. We needed better tools, a new approach.
And that's where this breakthrough comes in. It sounds like they really needed an out-of-the-box idea. How did they finally crack it?
Well, it was a big collaborative effort, led by CIRAD. That's the French Agricultural Research Center for International Development. They published their findings in Horticulture Research.
And what did they do?
It was ambitious. They looked at over 2,700 different triploid banana hybrids, and they genotyped them extensively, looking at over 200,000 SNPs.
SNPs, those are like tiny variations in the DNA code, genetic markers.
Exactly. Single nucleotide polymorphisms. They act like signposts across the genome. But the real innovation wasn't just the scale, it was the analysis.
Okay.
They developed a new statistical model. They call it the KC model. And here's the clever part. Instead of trying to untangle those messy, rearranged chromosomes,
Yeah.
they essentially filtered them out when estimating the genetic relatedness, the kinship between the plants.
Uh-huh. So they ignored the really scrambled parts of the instruction manual to focus on the clearer sections.
Sort of, yeah. They realized those rearranged regions were messing up the calculations used to find gene trait links. By excluding them specifically from the kinship part of the analysis, they could reduce the genetic noise.
Okay.
And this significantly boosted what they call the discovery power. Suddenly, they could see genetic signals, links between genes and traits that were completely hidden before because of all that chromosomal chaos.
Wow. So they cleaned up the signal by filtering out the noise from those rearrangements.
Precisely. It let them get a much clearer view of the banana's underlying genetic architecture.
So what did they actually see with this clearer view? What did the K8 model uncover?
The results were pretty striking. They identified a total of 62 QTLs, quantitative trait loci.
QTLs, those are regions of the genome that influence specific traits, right?
Exactly. And these 62 QTLs were linked to 23 really important traits for bananas. Things affecting yield, the plant structure, fruit quality like fruit size, how the bunch hangs, how long it takes to mature.
And these were new discoveries.
Many of them, yes. QTLs that conventional GWMS methods likely would have missed entirely. They simply hadn't been mapped before in bananas because of those genomic hurdles.
That's huge. Did they learn anything about where the good traits came from, like from wild ancestors?
They did, actually. They found strong links back to certain ancestral groups, especially one called the Banksy group.
Banksy.
Yeah. It seems this group contributed some really beneficial versions of genes' alleles for things like faster growth cycles and heavier fruit. It highlights how important wild relatives are as a source of useful genes.
That makes sense. Go back to the source.
And another really interesting thing they found was genomic hotspots.
Hotspots, meaning?
Meaning single regions on a chromosome, single QTLs that actually influence multiple different traits. It's a phenomenon called pleiotropy.
Okay, give me an example.
Sure. They found a QTL on chromosome 3 that affected both the weight of the fruit and the angle the bunch hangs at. Another one on chromosome 4 influenced both the time to maturity and how long the leaves persisted on the plant.
Huh. So one genetic tweak could potentially improve several things at once.
Exactly. That opens up possibilities for what breeders call multi-trait selection. You can potentially select for multiple improvements simultaneously by targeting these hotspot regions.
That sounds much more efficient. It really connects with what Dr. Guillaume Martin, the lead author, said, right? About turning the obstacle of rearrangements into an opportunity.
Absolutely. He basically said, look, these rearrangements clouded our view for ages, but by understanding them and adapting our tools because of them, we could finally see these crucial gene locations.
Yeah.
And he made the point that this isn't just about bananas, this methodological approach.
Right.
It could be a blueprint for tackling similar problems in other crops with complex rearranged genomes.
That's a really key point. If it works for bananas, maybe it works for, I don't know, other complex crops facing breeding challenges.
It certainly opens that door. It makes you wonder what else is hiding in plain sight in other plant genomes, just waiting for the right analytical key.
Okay, so let's get practical. So what? How do these discoveries actually help breeders on the ground? What tools does this give them?
Well, it gives them a much clearer genetic roadmap. They can now screen potential parent plants much more effectively.
How so?
They can look for those specific beneficial alleles, especially the ones traced back to that Banksy group, and select parents that have the best genetic profiles for the traits they want. It makes the selection process much more targeted.
Instead of just crossing fingers and hoping for the best after years of growing.
Pretty much, yeah. And it allows for early genotyping. They can test young hybrid seedlings genetically and predict their potential much earlier.
Ah, so they can weed out the unpromising ones before spending years and resources growing them out in expensive field trials.
Exactly right.
Yeah.
It massively streamlines the breeding pipeline. Saves time, saves money, speeds up the whole process of developing better varieties.
Makes sense.
The authors also suggest setting up pre-breeding programs. The goal there would be to create parent lines that are structurally homozygous, meaning their chromosome structures are more uniform, less rearranged.
Why would that?
It would likely improve genetic recombination, that shuffling of genes when you cross them, and it could also reduce genetic drag.
Genetic drag,
Yeah.
like accidentally bringing along bad traits linked to the good ones.
Precisely. So it's about making future breeding efforts even more efficient and predictable. The whole suite of tools and strategies coming out of this one study.
It really sounds like a potential game changer for banana breeding. But zooming out again, you mentioned this case model could be bigger than just bananas.
I really think so. The model itself, the approach of accounting for chromosomal rearrangements in this way, that's the key takeaway for other crops.
So any crop where breeders are hitting a wall because the genome is complex and rearranged, this offers a new way forward.
It provides a valuable framework, yes. A potential way to unlock trait mapping where it was previously blocked. It could accelerate the development of more resilient, higher-yielding varieties in lots of different species.
Which ties directly into global food security, doesn't it? Developing better crops faster is critical.
Absolutely critical. Especially with climate change and growing populations, we need crops that can withstand new pressures. This kind of research, solving a specific problem like banana genetics, can have these really broad, positive ripple effects across agriculture.
It's fascinating how solving one puzzle can provide tools for so many others.
It really is a testament to innovation in genetics.
So let's wrap this up. The core insight here seems to be that by creating genetic tools specifically tailored to the unique, messy reality of the banana genome, scientists didn't just unlock secrets for that fruit, they potentially forged a key for improving a whole range of other complex crops too.
That's a great way to put it. Overcoming a specific challenge led to a broader methodological advance.
So, it leaves us with a thought, doesn't it? Thinking about the future of food. If traits that were essentially invisible in bananas can now be seen and used, well, what other agricultural puzzles, things we thought were maybe impossible to solve, might just need a new way of looking? A different kind of genetic lens, perhaps? And how could rethinking those tools fundamentally change what ends up on all of our plates in the future?
That's the big question, isn't it? What other genetic potential is out there just waiting to be uncovered? Makes you think.